day. This is the Professor, CBMD PhD. And today's Friday, September 1st, 2023. It is 4:28 and 26 seconds, AM Eastern Standard Time. And today's research, avoiding cancer, longevity's gene. So let's hum to it here. Longevity gene from pagan mole. Rats extends. Lifespan of mice. The successful transfer of a gene that produces HMWHA paves the way for improving the health and lifespan of humans too. Date August 23rd, 2023. Source, University of Rochester. Naked mole rats known for their long lifespans and exceptional resistance to age-related diseases have long captured the attention of the scientific community. By introducing a specific gene responsible for enhanced cellular repair and protection into mice, the scientific investigators and microbiologists have opened exciting possibilities for unlocking the secrets of aging and extending human lifespan. The study provides a proof of principle, a unique longevity mechanisms that involves the long-lived mammalian species can be exported to improve the lifespans of other mammals. The researchers say they successfully transferred a gene responsible for making high molecular weight, hyaluronic acid, from a naked mole rat to mice. This led to improved health and an approximate 4.4% increase in median lifespan for the mice. Naked mole rats are mouse-sized rodents that have exceptional longevity for rodents of their size. They can live up to 41 years, nearly 10 times as long as similar-sized rodents. Unlike many other species, naked mole rats do not often contract diseases, including neurodegeneration, cardiovascular disease, arthritis, and cancer, as they age. The scientific investigators and microbiologists have devoted decades of research to understanding the unique mechanisms that naked mole rats use to protect themselves against aging and diseases. As compared to mice and humans, naked mole rats have about 10 times more HMWHA in their bodies. When the researchers removed HMWHA from naked mole rat cells, the cells were more likely to form tumors. The team genetically modified a mouse model to produce a naked mole rat version of the hyaluronic synthase 2 gene, which is a gene responsible for making a protein that produces HMWHA. While all mammals have the hydrolonium synthase 2 gene, the naked mole rat version seems to be enhanced to drive stronger gene expression. The researchers found that the mice that had the naked mole rat version of the gene had better protection against both spontaneous tumors and chemically induced skin cancer. The mice also had improved overall health and lived longer compared to regular mice. As the mice with the naked mole rat version of the gene aged, they had less inflammation in different parts of their bodies, inflammation being a hallmark of aging, and maintained a healthier gut. While more research is needed on exactly why HMWHA has such beneficial effects, the researchers believe it is due to HMWHA's ability to directly regulate the immune system. The findings open new possibilities for exploring how HMWHA can also be used to improve lifespan and reduce inflammation-related diseases in humans. It took them 10 years from the discovery of HMWHA, the naked mole rat, to showing that HMWHA improves health in mice. The next goal is to transfer this benefit to humans. They believe they can accomplish this through two routes, either by slowing down degradation of HMWHA or by enhancing HMWHA synthesis. They already have identified molecules that slow down hyaluronic degradation and are testing them in preclinical trials. The scientific investigators and microbiologists hope that their findings will provide the first, but not the last, example of how longevity adaptations from a long-lived species can be adapted to benefit human longevity and health. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and Leader of Research This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.